Let's look chemically at how this peptide bond is going to form. Once the tRNAs get placed into the proper location in the ribosome, then a peptide bond is going to form between the two amino acids in those two different sites of the ribosome. So if the first amino acid that came in was methionine, and we know that AUG always encodes that methionine, it has an amino group, it has a carboxylic acid group, and of course then it's going to have some sort of functional R group sticking off of it. The next amino acid coming in, let's say, is tyrosine. Tyrosine is going to have an amino group, a carboxylic acid group, and of course a functional R group. There has to be a reaction between the carboxylic acid of the methionine and the amino group of the tyrosine. When that reaction occurs, there's basically a dehydrogenation reaction where, dehydration reaction, where a molecule of H2O is released from this, and now we have a very strong peptide bond. This peptide bond is very strong. It helps hold the backbone of a protein together so that the amino acids are stuck together very tightly. And this is actually what dictates the primary structure of a protein. The primary structure of a protein are these very strong peptide bonds that hold the amino acids together. So, in the end, now we have a dipeptide shown here with methionine and tyrosine linked together through this very strong peptide bond. This process will occur over and over and over again as a protein builds itself, so that in the end, you're going to have an oligo and a oligopeptide, and then eventually a very long protein structure. Something you need to remember is the name of the enzyme that catalyzes this peptide bond formation. And the name of that enzyme is peptidyltransferase. Peptidyltransferase is going to catalyze the formation of these peptide bonds. Where is the enzyme's activity associated with peptidyltransferase? It's actually a tricky point. Peptidyltransferase isn't a protein enzyme like 99.99% of other enzymes. Peptidyltransferase is an example of a ribozyme. Now you're thinking to yourself, what is a ribozyme? A ribozyme is actually an RNA species that has catalytic activity. So we'll see in a little bit that peptidyltransferase activity is associated with the large ribosomal subunit. So the large ribosomal subunit can help catalyze the formation of this peptide bond. Now, let's take these amino acids and put them now into the context of the ribosome. There's two different sites of the ribosome. There's going to be a P site, which stands for the peptidyl site. It's the site on the ribosome which is holding the growing peptide. There's also an A site. The A site stands for the attachment site. It's where each new transfer RNA molecule attached to an amino acid is going to come into the ribosome. So it's the amino acid attached to tRNA in the P site. You see that this is a tRNA that encodes methionine, and it's actually attached to methionine. Along comes a transfer RNA encoding tyrosine, and thankfully, aminoacyl tRNA synthetase has done its job, and it's attached tyrosine to this tRNA. So this is coming into the A site, and now, although you can't visualize it on this slide, peptidyltransferase is going to catalyze this reaction with the carboxylic acid of the methionine and the amino group of the tyrosine. There's going to be a peptide bond that forms, and immediately now the ribosome is going to shift. It's going to shift so that what was once in the A site, this aminoacyl tRNA attached to tyrosine, is now going to be in the P site. So now the A site will become the P site. That's how translation is going to operate. Let's now take a look more specifically at the mechanism of translation as to how it works in the context of the ribosome. So we're going to look at an example and we're going to pretend that we're a prokaryotic cell. So we're in the confines of a little bacteria, and we're going to initiate translation. Now, the way the translation is initiated in bacteria is binding of the small ribosomal subunit to the messenger RNA. Can you recall what the small ribosomal subunit in bacteria is referred to as? It's the 30S ribosome. And what is the sequence to which the 30S ribosome binds to on the messenger RNA? It's the Shine-Dalgarno sequence. Now, having said that, what do we refer to a small eukaryotic ribosomal subunit as? Well, that would be the 40S. Remember, the prokaryotes begin with an odd number, meaning a 30, and then the eukaryotes begin with an even, 4 for 40. Let's take a look. So we have a small ribosomal subunit, and in prokaryotes, this is a 30S ribosomal subunit. It's going to bind to a messenger RNA, and it's going to bind to the messenger RNA at the Shine-Dalgarno sequence. The 40S in eukaryotes, the 40S ribosome, is going to bind to the cap. If you recall, prokaryotic messages are not capped, and so there's no way for a prokaryotic ribosome to bind to a cap. In the same way, there's no Shine-Dalgarno sequence in eukaryotes, so there's no way for the 40S ribosomal subunit to bind to a Shine-Dalgarno sequence. So the binding is going to now initiate translation. And what's going to happen is the first codon is going to always encode AUG, and a transfer RNA that's specific for that codon is going to come in carrying methionine with it. So we now have the small ribosomal subunit associating with the messenger RNA. We have a transfer RNA corresponding to the first codon, AUG, and now we have a large subunit that's going to come into play. In the prokaryotes, what do we call the large ribosomal subunit? We call it the 50S. In eukaryotes, what do we call the large ribosomal subunit? We call it the 60S ribosome. So some GTP energy is going to be burned in this process, and now we're going to get initiation at its maturest form. We have a small ribosome, a large ribosome, we have a messenger RNA, and we have the transfer RNA corresponding to AUG that's come into play. At this point in time, translation initiation is completed, and we can start building the protein. Notice something interesting about translation initiation. It's the only time during our translation machinery where the incoming amino acid is going to attach to the P site. At all other points in translation, the incoming amino acid is going to come into the A, or attachment site. So the only time the incoming amino acid binds to the P site is at initiation. A couple of other key points. Number one, in eukaryotes, it's methionine attached to the tRNA that's going to come into the P site. One way we can differentiate prokaryotic translation from eukaryotic translation is that it's formylated methionine. So there's going to be a little formyl group attached to the methionine when we're thinking about prokaryotes. That's a slight subtle difference between prokaryotic and eukaryotic translation. Here's a good point for you to remember the fact that specific antibiotics are going to bind to and inhibit translation initiation. Let's take a look. If you remember, aminoglycosides like streptomycin, like neomycin, like gentamicin, these are going to bind to, and they're going to bind to the small ribosomal subunit, the 30S ribosomal subunit, and they're going to inhibit initiation of translation. That's a key point to remember for the board exams. Another key point to remember is the following. What happens if you get a case that says a patient had a bacterial infection? They were administered a certain antibiotic that functioned in a certain way. And all of a sudden they started getting side effects from this antibiotic. Well, you're going to need to put together the side effects with the antibiotic, with its mechanism of action. So for aminoglycosides, the classic side effect is what? It's ototoxicity, toxicity up here in the ears, and it's also nephrotoxicity, toxicity back in the kidneys. So aminoglycosides have those two major side effects, particularly when they're used in combination with what? With a loop diuretic. That's a classic question. Now, for biochemical purposes, you need to remember that aminoglycosides are going to specifically inhibit translation initiation by binding to the small 30S ribosomal subunit of bacteria. The next step in translation, after initiation is complete and the aminoacyl tRNA attached to methionine is attached to the P site, the next step is elongation. So how does elongation happen? Elongation is going to happen when the incoming amino acids are going to start attaching to the A site, and the ribosome is going to shift down. And then a new A site will open up, an amino acid will come in, and the ribosome will shift down, and a new amino acid will come in. This process requires several cofactors. It requires some GTP energy. Prokaryotes are going to have elongation factors that are required. In eukaryotes, there's going to be little e standing for eukaryotic elongation factors required. You don't have to remember these for the board exam, so don't worry about it. As long as you remember that there's some energy required, there's some elongation factors that come into play, that's going to be good enough. There's one specific elongation factor later on that you'll have to remember. Now, the elongation of translation is going to be inhibited by another antibiotic. It's going to be inhibited by tetracycline. How is tetracycline going to function? It's going to bind to, once again, the small ribosomal subunit, and it's going to block entry at that A site. So, when tetracycline is on board, it's going to bind to the bacterial ribosomes, the 30S subunit, and the A site is going to prevent new transfer RNAs from coming in to elongate the protein. That's tetracycline and doxycycline and minocycline and any other related drug is going to operate in that fashion. Now the peptide bond is going to form. If you remember, there was an enzyme called peptidyltransferase. Peptidyltransferase had ribozyme activity that was associated with the large ribosome. The large ribosome is going to use energy from high energy bonds. You don't need to worry about that other than to say that high energy bonds are required. And the high energy bonds are going to form cause the enzyme activity. The breakage of those high energy bonds are going to allow the high energy peptide bond to form between methionine and leucine in this case. So now, what was once in the P site here, this aminoacyl tRNA attached to methionine, is now going to covalently bond with leucine. And now what you see is that in the A site, you have a dipeptide of methionine and leucine. As soon as that happens, the ribosome is going to shift down. And now, what was once in the A site, this dipeptide, is going to be present in the P site. So the P site will hold the tRNA attached to the growing peptide. That's how elongation happens. What's an important antibiotic that functions here? It's chloramphenicol. Not very frequently used in the US anymore due to some toxic effects, but chloramphenicol is a good inhibitor. It blocks elongation of translation. How does it block? It blocks by inhibiting peptidyltransferase. So chloramphenicol would then bind to the large ribosomal subunit and block peptidyltransferase activity. So, the third step, which we already alluded to in elongation, would then be the translocation step. So what was once in the A site is now, after the ribosome moves, is going to be in the P site. Methionine attached to leucine is now going to be in this P site. And so the A site is open, the codon is available for a new transfer RNA and its anticodon to base pair, and bring in with it a new amino acid. Translocation is something we'll spend just another second talking about. It's also going to require some energy. There's certain factors, elongation factors required. The one elongation factor that you should commit to memory is this one down here. It's eukaryotic elongation factor 2. Eukaryotic elongation factor 2. I'll explain why in a second. If you notice over here, we've boxed in here the macrolide antibiotics, such as erythromycin, or azithromycin, or clarithromycin. Any of these macrolide antibiotics, commonly used nowadays, these work by blocking translation elongation. Now, how do they block translation elongation? They're going to do so by inhibiting elongation by binding the 50S large ribosomal subunit. So that's how these are going to function. Now, getting back to elongation factor 2, I want you to recall that there's going to be a couple of bacterial toxins which inhibit that particular elongation factor. Which bacteria have those toxins? Can you remember? Well, there's two big ones. There's Pseudomonas aeruginosa toxin, and there's also Corynebacterium diphtheria toxin. Let's look. These toxins going to bind to and inhibit elongation factor 2 through a special mechanism, one that you should be familiar with. It's called ADP ribosylation. Okay, ADP ribosylation is when one of these toxins secretes, or I should say one of these toxins has an enzyme activity, one of the toxins is secreted, and it has an enzyme activity that causes an ADP ribosyl group to be attached to one of your elongation factors. The most important clinically is elongation factor 2. Now, when elongation factor 2 gets ADP ribosylated, that's going to inhibit its activity. So what's the net result? You're going to lower host protein synthesis. And now all the amino acids are going to be available for bacterial synthesis. So this is a way the bacteria, they prevent our cells from growing and dividing and functioning, and they allow themselves to propagate. Okay, so don't forget this ADP ribosylation step, and it's present in diphtheria and Pseudomonas toxin, and what gets ADP ribosylated is eukaryotic elongation factor 2. There's actually a couple of other places you're going to see ADP ribosylation. There's G proteins that get ADP ribosylated. So later on, when you look at G protein function, don't forget that certain toxins, such as Vibrio cholera toxin, enterotoxigenic E. coli toxin, even Bordetella pertussis toxin, these can function through ADP ribosylation as well. Those don't affect translation, however. Those are going to affect G protein function. So don't confuse the two. They work similar through the same mechanism, but they affect two different processes in the cell. The last step in translation is the termination step. The terminator, right? Okay, what's going to cause translation to terminate? It's when a stop codon comes into the A site on the ribosome. Can you remember what the stop codons are? You are gone, you go away, you are away. So in this case, a UAG codon is going to come into the A site, and when that happens, there is no amino acyl tRNA to come in. Instead, what's going to come in is a stop factor. A stop factor is going to come in, it's going to bind right here, and that's going to cause the ribosome to be released from the mRNA, to be released from the protein, and what you now have is a newly synthesized protein. There's some GTP energy required here, which you don't have to concern yourself with. The exam is not so concerned with your ability to count GTPs and ATPs. It's more interested in your ability to synthesize and integrate the information you've learned in your first 2 years of medical school. So here's a fully synthesized protein from its amino terminus to its carboxy terminus. It's now going to be modified within probably the confines of the endoplasmic reticulum or directly secreted from the cell. It just depends on the protein. So once the protein synthesis is completed, the ribosomal subunits are going to separate and the mRNA is going to be released. Not very complicated. I want to just remind you of a few abbreviations that are commonly used and perhaps might be used on test questions. In the process of translation, you see that EF and IF are used. EF stands for elongation factor. IF stands for initiation factor. And what does TF stand for? It stands for transcription factor. So TF is something not used in translation. It's something that gets used in transcription. So TF, IF, and EF. Abbreviations that might be used, I want you to be familiar with those.